Hi everyone, this is so exciting. This is our last week of genetics. So we're going to be talking about sex-linked traits and pedigrees. So when we're talking about sex-linked traits, we're actually talking about the traits that are on the X sex chromosomes. So these traits can be dominant or recessive. The probability of inheritance is altered because the trait is on the X chromosome. So what that means is if a male ends up getting the trait, they automatically have it because they only have one X. So a lot of the times you will see females will be carriers, so they won't have the trait, but they'll have one allele for it, and they then would pass it on to their sons. They're called carriers. So they're technically healthy and don't have the trait, but they'll pass it on to their sons, and then the sons will get it. So the traits we're going to be referring to is like colorblindness, hemophilia. So when you're writing your genotypes, you must write XX for females and XY for males. So you need to put the sex chromosomes in there. And then what ends up happening is you actually put the alleles on top of the X's. So they're actually superscripts on top of them. So when we're talking about sex-linked traits, we're referring to on the X's again. The two traits that we're going to be referring to are colorblindness and hemophilia. So what that means is you can have a male that has colorblindness or a male that has no colorblindness. Those are the only two options in your phenotypes. Male with it, male without. Then females, they can be completely healthy, meaning homozygous, or they can be carriers. So it's really important when you're writing your phenotypic ratios to say whether or not the females are carriers. Because if they're carrying, say, hemophilia, which is actually a blood disorder, it's when individuals' blood doesn't clot like normal. So potentially anybody that has hemophilia, if they get a little cut, just like a paper cut, they could potentially bleed to death because they don't form scabs like we do. They don't get the clotting factor in their blood. So it's important to say whether or not a female is a carrier, because those females will want to know what their chances of having a child that has this deadly disease. And then you can have females that have it. So there's females that have hemophilia. They would end up having to have two of those alleles on their X's. So on each of the X's, they would get the alleles. Now, colorblindness and hemophilia are both recessive traits, so you're still going to use lowercase letters when you're referring to those. So here's an example of a Punnett square problem dealing with hemophilia. So they'll tell you in the problem that it's recessive. So capital H would stand for normal. Lowercase h would be hemophilia. They want you to cross a normal male. So if he's normal, he's going to have one X. So he gets the X and the Y genotype, and then you would put the capital H on top of the X. So it's called a superscript. So then you're going to cross the normal male with a female with hemophilia. So if she's a female and she has hemophilia, well, hemophilia is recessive. That means on each of her X's, she would have to have the lowercase h, okay? So then what you do is you put the X's and the Y's together, and the superscripts come with it. So in this case, this would be a female. She's healthy or normal, but again, she is a carrier. So if you see the heterozygous form for normal in females, these are carriers. So you're going to want to name and make sure you say they're carriers. This male then would have hemophilia. So you go up to the top, lowercase, he's recessive, hemophilia. This female, again, she's a carrier. So she's healthy, but she's a carrier. And then here's a hemophilic male again. Okay, so your genotypic ratio would be 2x big H, x little h to 2x little h, y. So then the phenotypic ratio, you would say 2 female carriers. You could even say 2 healthy females, but they're carriers. You got to say they're carriers because it's a disease. And then 2 hemophilic males. Now, the probability problems are a little bit tricky. Sometimes the problems will just ask, what's the probability of having a hemophiliac child? Okay, so if they're talking about the whole children, you're going to put your answer out of the 4 boxes. So in this case, the probability of having hemophilia would be these 2. So you would say 2 out of 4, 50%. Now, some of the questions for probability will ask, well, what is the chance of just having a male child with hemophilia? So they're just referring to the male children. you just want to look at the males. So in this case, it would be 100% for hemophilia because it's 2 out of 2 and they both have hemophilia. So you would say 100%. Now, maybe they ask for the females. What's the probability of having females, children with hemophilia? You just look at the females. So there's 2 of them. Well, in this case, none of them have hemophilia. They're just carriers. So they're totally healthy because capital H dominates lowercase h. But they are carriers. So they do not have hemophilia. So you would say 0%. There's embedded videos that I would like you guys to watch, and also make sure you're watching the sex-linked video tutorial on how to do the problems. That will also give you more problems to look at. So our last genetic topic is pedigrees. So with pedigrees, basically is like a family tree of all members that have a trait. Okay, so it's a chart that shows you and it tracks which members of the family actually express the trait. So whether we're talking about dimples or we're talking about hemophilia. So the males are always represented by squares. So these are the males. This is a male. Females are always represented by circles. Now, if you see a line between 2 individuals like this, that means these 2 people are married. The lines going down represents these are the children of these individuals. Okay? Now, if you see something shaded in, that means this individual has the trait that we're talking about. So if we're just looking at this little Punnett square, only this female has the trait. Now, sometimes in pedigrees, they will represent half-shaded squares or circles. That means that person is a carrier or heterozygous. But often they don't do that, and you guys actually have to go through and figure out if the individuals are homozygous dominant or are they heterozygous. Now, if it's recessive, then they're totally homozygous, always. Now, when you're interpreting a pedigree, the first thing you need to do is you need to figure out if it's autosomal or is it sex-linked. So this is the rules for whether or not it's autosomal or sex-linked. So if most of the affected individuals, meaning those individuals that have the trait, are males, then it's most likely sex-linked, okay? Because more males end up getting it because they only have one X. If there's more of a 50-50 split or close to it, the ratio between like males and females, a 50-50 split, then it's most likely autosomal. So that's going to be important when you're going to have to go back and write the genotypes, because if it's autosomal, you can just use normal letters like capital D, lowercase d as the genotypes. But if it's sex-linked, you have to put the X and the Y and the X and the X and then put the alleles on the X's. So that's the rules for figuring out if it's autosomal or sex-linked. Then the next thing you have to figure out, is it dominant or is it recessive? So if it is dominant, at least one parent must have the trait, and it will be seen in every generation. If it's recessive, you may see 2 parents that don't have it, but then they have a child that does. So you're going to see that it'll skip generations. That means it's recessive. So that's where you would use your lowercase letters for recessive, and dominant would get the capital letters. So before we figure out if it's autosomal or sex-linked or dominant or recessive, if you just generally look at this pedigree, you're going to see it has 4 generations. So they'll number the generations often with Roman numerals, okay? So it would be the first generation would just be these 2, then these would be the second generation, third generation, and then down here would be your 4th generation, okay? Sometimes they'll number the individuals in each generation, or sometimes they'll just number some of them if they want you to identify just some of the genotypes of the individuals. So you may get general questions about pedigrees, like how many females are there? So remember, females are the circles. You just count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there will be 9 females. Then they may ask you, how many males are there? 1, 2, 3, 4, 5, 6, 7, 8, 9 males. They may ask you how many individuals are affected or have the trait. You would say 1, 2, 3, because only 3 are shaded. Then sometimes they'll ask you like how individuals are related. Okay, so for example, like how is this male, number 1, related to this male? Well, they're actually cousins. So these two are cousins. Then they might ask you, like, what is the relationship between this girl and this female? Well, this little girl is the granddaughter of this individual. Okay, so there's just like general questions like that you will also be asked. Now, again, if it's autosomal or sex-linked. So autosomal, you have to go back to your rules. Is there a 50-50 split between males and females that have it, or close to 50-50 split, or do mostly males have it? Well, in this case, mostly males have it, so it would be sex-linked. Okay, so then the next thing we need to figure out, is it dominant or is it recessive? So again, if it's dominant, you're going to see at least one parent have it, and in every generation. If it's recessive, you're going to see two parents that don't have it, but then they have a child that does. So in this case, or it skips generations as well. So in this case, you see you have two parents here that don't have it, but they have a child that does. Same thing here. Don't have it, child that does. Same thing here. Don't have it, child that does. So in this case, this would be recessive. Now, next thing you might be asked to do is write the genotypes for these individuals. So in that case, for anybody that's shaded in, so the males here, if it's sex-linked, you guys would have to write XY for the genotype, and then you would have to put the lowercase allele because it's recessive. So say we're talking about hemophilia, because that's sex-linked recessive, you would have to put the little h on the X. So it would be X, little h, and then Y for all these males. And then all the males that aren't shaded would be X, big H, Ys. So all these people, these males, would be X, big H, Y. Then you got to go back to the females. So all these females, they don't have hemophilia. So they're not going to have the genotype X, little h, X, little h. However, you are going to need to go back and figure out if they're carriers or not, okay? If you're not quite sure, what you can do is you write X and then you put a big H, and then the other X, you put a question mark, or you can end up writing the two genotypes totally separate. You would be like X big H, and then next to it, like slash, you would do X big H, X little h. So you actually end up writing both of them. I'm going to post another video tutorial, and I'll make sure to write that down so you guys can see what actually I'm like referring to when I'm talking about that. All right, so here's our next one. So autosomal or sex-linked. So again, autosomal, there's more of a 50-50 split between male and females. Sex-linked, mostly males have it. So in this case, we have a 50-50 split. So it would be autosomal. And is it dominant or recessive? So again, dominant, you're going to see it in every generation. At least one parent will have it if they have a child that has it. And recessive, it would skip a generation, and you would see two parents that don't have it with a child that does. So in this case, we have dominant. Okay, so you're going to see it's every generation, the trait, and at least one parent has to have it if they have a kid that has it. So that's what we have here. Okay, so if we are writing the genotypes for these individuals, every individual that's not shaded in, we would give them the homozygous recessive genotype. So say we use D's, the letter D. All of these individuals that are not shaded in would all get lowercase d's. So 2 lowercase d's. And then these individuals, again, you would have to go back and you have to figure out if they were heterozygous or homozygous dominant. So in this case, this female, she would have to be heterozygous, so have a big D, little d, because she has children that have 2 little d's. Okay, this one's not, because this one's married in. And again, if you're not completely sure if it's homozygous dominant or heterozygous, you could write your genotype as capital D and then question mark, because you don't know if it's capital D, or capital D, lowercase d. But if you want to write both of them just to be safe, you can do that also. I hope this helps. If you guys have any questions, please email me.